any point you have questions, please put them in the chat. So your exam is going to be unlike every exam that you've taken so far here. It's going to be set up so that it is mostly multiple choice. So at 9:05, you will log onto Canvas. You will then have 35 minutes to answer 45 multiple choice questions. The questions will be open from 9. I think I opened them a little early, so I think you can get on at 9, but technically from 9:05 to 9:40 are your 35 minutes. You will then go to the second part, and I'll walk you through Canvas and show you how that works, and you'll download the short answer document, and you will have that available from 9:40 until 10 o'clock, okay? It's 2 short answer questions, and what you will do is you will submit it through Canvas. So please don't email me all your short answer questions. You're going to submit it through Canvas. And what Canvas will do is Canvas will check it for plagiarism. So what that means is that if you copied and pasted it from Google or the internet or whatever, it will come back with a plagiarism score. And I will use that to make sure that everyone is just writing what they think. Okay. The test is designed so that you will only see one multiple choice question at a time and that you can't go back. So what this prevents and the rationale behind this is this makes it so that if 2 people happen to be sitting next to each other and they're both taking the exam, they can't work together on it because the questions are also randomized. So everyone will see a different order of questions, and you'll have to answer that question and then you'll move on. I want you to take this like a regular test. So I am not making you use the proctoring software because it costs money, and that doesn't seem fair right now. So I'm not making you pay for anything. I'm just going to trust that you're not going to use your notes, you're not going to use your book, and you're not going to use your phone. I'm going to trust that you're going to take this like a normal test. Okay. So with that being said, I'm going to show you where you can find all of this stuff briefly. Okay. So if you are in Canvas, here's Canvas for you guys, right? If you go to assignments, you'll see your upcoming assignments. So as a reminder, that muscle worksheet is due in less than an hour. That should have been submitted. Muscle questions for your discussion board points are due, I read that at 8 a.m. tomorrow, so you have to answer that. And then this will be available. So this will be available, your multiple choice questions, and then your short answer questions will be available. You click on it, this assignment will lock. It'll tell you what to do, okay? The other thing I want to draw your attention to here is in quizzes. In quizzes, I put up a technology survey that I would like you to answer. It's ungraded and it just tells me, have you been able to access WebEx? Yes or no? Is it going okay for you? Are you looking at the videos on YouTube? Is that going okay for you? So please just take that survey. Okay. I'm now going to switch gears here, and if I could figure out, I'm going to stop sharing this. Okay. Are there any questions about the format of the exam? If there are, please put them in the chat window below. And twice. Okay, I'm going to now go through the questions I got for the review session. Okay, so share content, and so much better at this whole thing here. Okay, so I got a few questions. We're going to go through them. I've also posted this to Canvas if you guys want to look at it that way. I'm going to make sure I still have the chat feature. Okay, so the major question that I got was about muscle contraction. And this question came in many forms. How does a muscle cell contract? How does a muscle fiber contract? Remember, muscle cell and muscle fiber, those are synonyms. They mean the same thing. How do motor neurons signal cells? How are the bands? Why are the bands of the sarcomere important? Why is the sarcomere important? What does acetylcholine do? What does ATP do? What does acetylcholinesterase do? What does calcium do? So I'm going to walk through all of this under the general heading of muscle contraction. Okay, so the skeletal muscle are fibers that contain myofibrils. So again, skeletal muscle cells, we call them muscle fibers. The plasma membrane, we give a special name, we call it the sarcolemma. These cells are filled with fibrils, which are what we call the cytoskeleton, the actin, and the myosin. Okay, and they have alternating bands of dark and light when you look at them. The dark bands we call A bands, the light bands we call I bands. And these myofibrils are made of sarcomeres. And sarcomeres are important because they're what does the contracting. So the sarcomeres are the cytoskeletal elements that do the contraction for muscles. They are what contracts, okay? You do need to know how to label the sarcomere, which is why I'm going through it now. So the middle of that I band is the Z disc, and the sarcomere goes from Z disc to Z disc, Z disc to Z disc. Okay. In the middle of the A band is a light area called the H zone, and in the middle of that is the N line. So you need to be able to draw this. I would recommend at the end of this class video that you take a minute and you draw a sarcomere. Pause it if you're watching this video at home. Draw a sarcomere. Okay. If we zoom in on the sarcomere, again, sarcomere goes from Z disc to Z disc, and the thin filaments are actin, and they're anchored to the Z disc, but they're not present in the middle, the H zone. The thick filaments are myosin. Myosin is what does the work, okay? Myosin is going to split ATP to generate the power needed to pull actin to the middle. Now, what is the I band and the A band, or that light area and the dark area? The light area is where there's only actin. So the light area is near the Z disc, where there's only those thin actin filaments. That's what makes it light. And the dark band, the A band, is the area where you will find the myosin filaments. Okay, that's what gives them their name. So you're going to be able to draw a sarcomere. Okay, let's now talk about how that sarcomere contracts. So keep in mind all these vocab words: excitable, contractible, extensible, and elastic. Muscles have to be stimulated by nerves to contract. Skeletal muscles have to be stimulated by nerves. These are voluntary, right? So we're going to think the thought, make a bicep flexion here, and we're going to then send a signal through our motor neuron to tell it to contract. Okay, keep in mind a motor neuron is one neuron, and all the muscle cells it stimulates, they meet at an area called the neuromuscular junction. And the motor neuron is going to release acetylcholine into that synaptic cleft, or the gap between the neuron and the muscle. These motor neurons come from the spinal cord. They come from the spinal cord, and again, they're going to form motor units where they innervate, where they signal to muscle cells or muscle fibers. And they release acetylcholine into that synaptic cleft. Acetylcholine is released because the nerve gets an impulse that's going to come down the nerve fiber. The nerve gets an impulse, it comes down the fiber. Calcium comes into the neuron, and the calcium tells the neuron, okay, dump out all that acetylcholine. The acetylcholine will dump out into the synaptic cleft. It'll float on down, and it will bind to the muscle cell plasma membrane, what we call the sarcolemma. This will open up a cation channel. Remember, cations are positive. Cats are good things in your life. You want to have a cat, okay? Cats are positive. Sodium is our speed runner. Sodium is our speed runner. Sodium is going to rush into the muscle cell fast. This is going to cause the muscle cell to become positively charged. Sodium is going to rush into the muscle cell. This is going to cause the muscle cell to become positively charged. We call this a depolarization. Then potassium is our slow tortoise type runner. Potassium is going to eventually leave the cell, but it's going to go slower. And that's going to cause the positive charge to move back out. We call this wave of positive charge moving in with the sodium and moving out with the potassium. As it moves down the muscle cell, we call it an action potential. Okay, a change in electrical signaling. How does this whole thing end? Well, it ends when the acetylcholine is broken down. And the acetylcholine is broken down by acetylcholinesterase that we abbreviate ACHE, acetylcholinesterase. Okay, so the acetylcholinesterase will stop the contraction. Again, that action potential will spread down the muscle fiber. So where the motor neuron is, that's where the signal will start, that wave of positive charge, and it will move down the muscle cell. The electrical charge will spread. Now, what does that positive charge do? That positive charge causes the muscle to contract. And how does the muscle contract? The muscle contracts via the sliding filament model, where those actin fibers are going to come together. And that area in the middle, that when the muscle is relaxed, we don't see any actin fibers, that's going to disappear because all the fibers are shrinking together. The action potential will cause the myosin heads to grab up, pull onto the actin, and pull them to the midline. And they'll do this several times. And what they need is calcium. So calcium is super important, not just for bones, but also for muscles, because calcium is responsible for the contraction. So where does the calcium come from? Calcium comes from the sarcoplasmic reticulum. At rest, those myosin heads can't grab the actin, it's blocked. And so calcium is what opens up the actin and allows the myosin to grab it. How does the calcium get released? The calcium gets released because of that action potential. So if we put it all in order, an action potential spreads down the muscle fiber, that action potential opens up the calcium stores from the sarcoplasmic reticulum. Calcium will fill the muscle fiber. That calcium allows myosin to grab onto the actin and pull it to the midline, contracting. And myosin will use ATP as the energy source. So, what are the two things that a muscle needs in order to contract? Well, it needs calcium and it needs ATP. The muscle needs calcium and it needs ATP. ATP provides the energy for contraction, and calcium opens up the actin so that myosin can grab it. And all of this comes from the structure of the muscle fiber, right? That calcium is coming because of that action potential. So the motor neuron releases acetylcholine. Acetylcholine opens up those cation channels. Sodium rushes in. Potassium slowly leaves. That wave of electrical change, that action potential, opens up the calcium stores in the sarcoplasmic reticulum. The calcium will then go to actin, open it up so myosin can grab it. Myosin will use ATP, pull the actin to the midline. This whole thing will go until the signal stops, until acetylcholinesterase breaks down all the acetylcholine, or the muscle runs out of ATP. ATP is a limited resource. Okay. Now, what I'm going to ask to keep the video consistent is to hold questions until the end. I'm going to go through all the questions I got. I'm going to stop the recording and then I'll open up the chat feature. So I'm going to keep going. I'm going to just keep talking. Okay. That way, if you watch this video back, you can pause it at several times. So the next question I got is, what is the difference between osteoblasts and osteoclasts? And I also got a few questions about the process of bone growth and bone remodeling. So I'm just going to go through that briefly. Ossification. So when we are in development as an embryo, eventually into a fetus, into a child, our skeletal system starts as cartilage. And the cartilage needs to be replaced by bone. We call the process by which cartilage is replaced by bone ossification. And most of that happens by the time we're born, except we have a few key areas where we still have cartilage. For example, we have the fontanelles in our skull, which stay as that cartilage so that the skull can be more malleable to fit through the birthing canal. And in our long bones, we still have epiphyseal plates that are present as cartilage that help the bones grow lengthwise. Okay. In the process of ossification, cells called osteoblasts, think of these as our builder cells, are going to cover the cartilage with a bony matrix. They're going to dissolve and digest that hyaline cartilage and replace it with bone. So osteoblasts are the builders. Blasts are builders. Bone growth is when the bones grow lengthwise from those epiphyseal plates, where they're going to add cartilage on top and replace the cartilage on the bottom with bone. Again, that's done by those osteoblast cells, those builder cells. Bones can also remodel, and in bone remodeling, bones are going to grow in areas where they need to. Why would they need to grow? Well, if you're using your muscles, you're putting a lot of force on the bone at the area of the muscle attachment. Now that we know muscles, we know all about muscle origins and insertions, right? Where areas where the muscle will join the bone. Well, those areas of bones are under a high amount of stress. And so the muscles cause the bones to say, hey, we should add more bone here to help deal with this increase in force. And that's done in the process of bone remodeling, again, by those osteoblast cells. But they also work with osteoclasts. Osteoclasts are our chaos cells. They destroy bone. And so the osteoclasts are going to take away some of the bone on the inside or in areas where it's not being used. And the osteoblasts are going to add it to the spots where there's a lot of force. Bones can grow in width as we use our muscles. Our bones, we say our bones are getting stronger. They're becoming thicker. And this we call appositional growth. Okay? So again, in bone remodeling, osteoblasts are going to add bone to the external face, and osteoblasts are going to reabsorb, osteoclasts are going to reabsorb bone or destroy bone, take it away from the internal side. And in this way, we're going to increase the circumference. And this process can also be impacted by growth hormones. Okay. Let's talk about calcium. So our bones need calcium in order to grow. The osteoblasts lay down a matrix that is full of calcium. If calcium is low, however, our body doesn't care about our bones. Our body is more concerned with the amount of calcium in the blood. And so they're going to use the bones as a calcium store, and they're going to destroy it as needed to maintain blood calcium levels. So when blood calcium gets low, the parathyroid is going to release parathyroid hormone. And this is going to turn on those chaos cells, those osteoclasts, to release the calcium from the bones. So that's the role calcium can have. And again, muscles can help us increase our bone thickness. So calcium and blood calcium concentration tells us the when of bone remodeling, especially when we're talking about destroying bone. And the muscles tell us where. Where do we need to add more bone? So low calcium will tell us take away bone. Muscles action and muscle exercise will tell us add some bone here. We need more bone. Now answer the third question. The third question is, how is ATP regenerated? So muscles need ATP to contract. The issue is that we only have enough ATP stored for a few seconds of exercise. And so we need to make more ATP from a DP, adenosine diphosphate. We need to make more ATP, the triphosphate, from the diphosphate. We have three ways of doing that. The first is to use creatine. So we can directly take and steal that phosphate group from creatine phosphate, and we can add it to adenosine diphosphate to make adenosine triphosphate, which is energy, right? The muscles need that ATP in order to contract. Why do they need ATP to contract? Myosin. Myosin is going to use the ATP to grab the actin. The issue with direct phosphorylation, the issue with using creatine, is that we still don't have enough. We have enough for about 15 seconds worth of action. So all the creatine in your muscles will only give you less than a minute of activity. And that's because each creatine phosphate group can only make one ATP. So it goes pretty quickly. You can also do anaerobic respiration, right? Make an ATP without oxygen. For this, we're going to use glucose, and we're going to break it down and eventually get 2 ATP per glucose molecule. The issue is that we're still only getting 2 ATP, and we're making a toxic byproduct here called lactic acid. And this lactic acid promotes a lot of muscle soreness, which is bad, but it'll give you about 40 seconds worth of energy. So you can get a burst of energy without taking in any excess oxygen. And so this is when you're working out, sprinting, for example, and at that last second you feel a burst, you push forward, right? You're not taking in enough oxygen for your muscles to be able to use that, but you can go for about 40 seconds. The most ATP comes from aerobic respiration. So this occurs in your mitochondria. You're going to make 32 ATP for every glucose molecule. And this is how we make most of the ATP we use. It's good because we don't use acid, or we don't build up any acid, and we're using oxygen to make ATP here efficiently. Okay. Some people want to know about the pelvic girdle, the function of the pelvic girdle, and how it's different in males versus females. Your pelvic girdle is made of 2 coxal bones, which are your hip bones. Now, don't confuse the word coxal with coccyx. Coccyx is your tailbone. That's part of your vertebrae, okay? The coxal bones are the hip bones. The coxal bones are made from a fusion of the ilium, the pubis, and the ischium. So your pelvic girdle are those two coxal bones, those two hip bones, plus the sacrum and the coccyx in the posterior side. These are large, heavy bones that have deep, deep sockets for our thigh bones, and their main function is to bear weight. They hold the weight of your body, but they also protect your reproductive organs, especially in females. And they're shaped in a specific way. We've got, again, the fusion of the ilium, the ischium, and the pubis. The acetabulum is that deep socket that we talk about. The difference between males and females is in their shape. And for females, you just want to consider childbirth. You want to think about babies when you think about the female pelvis. And the female pelvis is shaped for childbirth. They've got a larger, skipped ahead. They've got more laterally flared ilia. They've got a shorter sacrum. They've got a larger inlet and a larger pubic arch to create a rounder, more spacious pelvic area to fit a child through. Whereas males, the opposite is true, okay? They're more vertically flared as opposed to laterally flared. They have a smaller pubic arch, a smaller true pelvis, okay? All of that is because they don't need to push a baby out. So they have more narrow hips than women do. Okay. I want to know about tetanus. Tetanus is a complete and fused contraction. So the muscle cell, each individual cell will contract or not, but the whole muscle can have a graded response. And so that can come from the number of cells stimulated, but you also want to make sure that the stimulation from the motor neurons is quick enough, is at a high enough frequency. If it just gets one stimulus from a motor neuron, one signal, that is what we call a twitch. So everyone's had those little eye twitches before, right? It's super annoying. That is just one motor neuron signaling onto that muscle fiber. What you want is a complete contraction, which we call a fused tetanus. This is where the motor neuron is firing fast enough so that they are all added together and the muscle will stay contracted. There's enough acetylcholine being released to make enough action potentials to release enough calcium to keep that contraction going. That is fused complete tetanus. If the motor neuron is not signaling fast enough, that is where you can have unfused tetanus, which is a shaking of the muscle, a reverberation. You see that a lot in people who have motor neuron diseases. So things like Parkinson's disease, for example, they'll have a characteristic tremor. That's because the motor neuron is not signaling fast enough because of damage. So again, the rapid stimulation will create a smooth, complete tetanus, whereas the force comes from the number of muscle cells that are contracting at once. Okay. One more person wants to know about muscle disorders. Muscular dystrophy, again, that's the inherited muscle destroying disease, usually diagnosed in childhood and fatal in their 20s and 30s, and it's caused by a breakdown in the sarcomere. So the sarcomere starts to fall apart. Whereas myasthenia gravis is adult onset, starts with muscle weakness, also fatal. The difference here is that it's not the sarcomere itself that's breaking down, it's the acetylcholine receptors. So the muscle is fine, it's just not getting the signal from the motor neuron, and that will eventually lead to death. Aging happens to us all. Muscles will atrophy, we call that sarcopenia. It's a decrease in muscle, a decrease in body mass, a decrease in muscle strength, but it can be maintained and rebuilt if you exercise effectively. So you want to maintain your muscles, use it or lose it. So I want to know about synovial joints. How do synovial joints move? So again, the plane joints, this is found in the intercarpals in your wrist. It's a sliding motion. It allows you to go like that. Spread your hands out. This is from plane joints, it's a flat surface. Hinge joints, think of your elbow. Uniaxial can only move like a hinge, move in one direction. Pivot joints, this is when they move over each other. The classical example here is your radial ulnar joint for that pronation supination movement. you need a pivot joint. Condylar joints, these are egg shapes. These allow for biaxial movement, so it's side to side and back and forth. You can't do a full 360. So these are our classic knuckle joints. You can't move your fingers in a full circle. However, you can get a little bit bigger of a circle in the saddle joint with our thumbs. So whereas our fingers move with a condylar joint, our thumbs have a little bit more movement with that saddle joint. So the carpal metacarpal joint of our thumb is a saddle joint. And the most movement comes from the ball and socket. So these are our hips and shoulders. They can move multi-axial all directions because of the shape of that joint. Okay. That was where the questions that I had gotten and received. So now what I want to do is open it up for questions. If you have questions, please put them in the chat feature. Otherwise, you are good to go. I will not see your faces Monday, but I will be around Monday for your exam, and I'm around this weekend if you have questions for the exam. Keep in mind, I am an old lady. I go to bed at 9:30. So if you ask me a question after 9:30, you are not going to get a response. I am asleep. Any questions people have? I'm going to end the recording and I'll stick around for another few minutes